Welcome everyone to today's webinar. My name is Marie Latulippe and I am the Director of Science Programs at IFINS, or the Institute for the Advancement of Food and Nutrition Sciences. So as we start, just a bit on IFINS, who is bringing you this dialogue today, but we are a nonprofit organization and we focus on advancing food and nutrition science for public benefit. And the way we do that is we bring together scientists from government, industry, and academia, and we do that to support collaboration and to catalyze knowledge. We also fund actionable research, and that research is conducted by external individuals who are selected through an open and competitive process. And if you're interested, you can find all of our currently funded projects on our website. We also advance food and nutrition science for public benefit by convening these experts across sectors to engage in a dialogue. And part of our requirement for funded researchers is to commit to peer-reviewed publication regardless of results, and we do fund open access of all of our work so that it's widely available. And then finally, we just like to note that we do conduct our work by leveraging pooled industry funding in combination with in-kind contributions from both the private and the public sectors. And for today's event, we just wanted to touch on the Gut Microbiome Committee of IFINS. And here you see the overall mission and focus areas of this committee. Today, this committee is kicking off a new mini webinar series that will be focused on the role of diet and gut microbiome in immune response and inflammation. And our intention here is to explore these relationships from different perspectives in order to understand the state of the science and gaps that could potentially be addressed through IFINS programs. So just a quick note on logistics. You should see this control panel on your screen. Basically, we encourage you to type your questions throughout the presentation, and we will be taking those at the end of the presentation. And for some of you, the control panel might look different like this, but still go to that question mark and type your questions in there. So with that, we are extremely pleased to jump into the program and introduce Dr. Emily Leeming. She is a dietitian researcher at King's College London, and she's senior nutrition scientist at Zoe. And her PhD thesis investigated novel and multivariate approaches to explore the impact of diet on gut microbiome. And Zoe is a personalized nutrition company that's actually running PREDICT, which probably many of you have heard of, and that is the world's largest nutrition study. And the purpose of that study is to understand the relationships between food and health. So Dr. Leeming will speak for about 20 minutes, and then we'll open it up for Q&A, and we will then also invite Dr. Sarah Berry to join us at that point. So with that, we turn the platform over to you, Dr. Leeming.
Hi everybody, and Marie, thank you so much for the introduction. It's great to be here and always excited to talk about diet and the microbiome. So that this gives me a fantastic opportunity. So today we're going to talk about kind of a brief overview of diet as a modulator of inflammation and immune response via the gut microbiome. What is the current state of the evidence, and what can we look towards in terms of dietary recommendations? So just for full disclosure, as Marie mentioned, I'm kind of senior nutrition scientist at Zoe, and Zoe is a commercial partner that funds the PREDICT studies, and I will be talking a little bit about the PREDICT studies very briefly later in this talk. So what is the gut microbiome? It is, considered our second brain, and it is a collection of microorganisms. Typically, we talk about bacteria, but really it is also a collection of viruses, fungi, and archaea. And these exist in the human gastrointestinal tract, so predominantly in the lower colon. And these are very much this, complex ecosystem that have been associated with both health and disease. So they've been shown to be associated with disease states across the body, such as heart disease, type 2 diabetes, NAFLD, which is fatty liver disease, metabolism and obesity, and also many interactions in terms of positive influences on your health. Primarily, their main objective is to digest and break down complex food molecules for producing energy, some vitamins and metabolites. But really what's our key interest here when we're talking about this is that 70% of our immune cells are housed in our gut. And really the microbiome is a key element. It trains the immune system to help us to fight pathogens. One key question that I get often asked is, what is a healthy microbiome? And I think it seems, that, there should be this recipe of a microbiome that we should be able to know is healthy or not. Sadly, we're not quite at that stage yet. What we do know is that it might not necessarily be about what's particular type of species that you have in your gut, but rather the function of those bacterial species themselves. So what happens is that these bacteria will consume and digest and metabolize food and produce a range of chemicals. And a certain number of species might produce the same chemicals, and it's these chemicals that exert a health effect, and these things that very much have this health profile. So really we think very much about the function rather than composition. Diet is a modifiable contributor to the microbiota. So it's thought to explain over 50% of the microbial structural variations in mice and 20% in humans. So very much signaling the potential for dietary strategies in disease management through the gut microbiota. And if we look more closely about how these interactions happen, we really see kind of a variety of effects. So you have your individual eating pattern, which you can see on the left, and we have this direct effect of diet on the host. And this is very much in the context that we know that around 90 to 95% of nutrients are actually absorbed in the small intestine, so before they actually reach the gut microbiome. Then we have the indirect effect of diet through the microbiome, which then has an effect on host health. And these 26,000 kind of food chemical compounds are metabolized by the gut microbiota and can perhaps be broken down into about 50,000 of those food chemicals that I mentioned, those kind of healthy chemicals, those metabolites, which can then be absorbed across the gastrointestinal barrier and to then exert a health effect. A key focus of the metabolites, and the ones we get particularly excited about and are very relevant to what we're talking about today, are the short chain fatty acids. And these are really the main metabolites that are produced by the gut microbiome, very specifically produced by fiber degrading microbes in the gut. Now these, we get particularly excited about butyrate, which is the kind of principal fuel for your intestinal epithelial cells, and they tend to exhibit kind of a range of anti-inflammatory properties, are very much helping you to support your gut intestinal barrier as well, which is a key part of supporting your immune system. If you think about your skin being external to your body, if you think that your gastrointestinal tract is this long tube, and actually it is also an interface with the external environment as well. So let's talk briefly about inflammation. So in broad terms, inflammation is your body's immune response to a stimulus. So this could be something like a common injury, like burning your finger or falling off a bicycle. And so then you get this affected area, which is perhaps, it feels very sore and it's red or puffy, and it's very much this localized injury, which, is actually represented by kind of increased blood flow and, capillary dilation. So in short, you kind of have this, very noticeable, typically, response where you're kind of fighting something that might turn out to be harmful. Well, we know, obviously, I feel like we need to all definitely acknowledge that actually, some form of inflammation is useful, because it's part of the fact that your immune system is actually working. However, when we have this low-grade inflammation, which is something that is unnoticeable by the host, means that we have this consistent kind of inflammatory response, which can then perhaps lead to problems in later life, so contributing to diabetes, cardiovascular disease, and autoimmune disorders. So inflammation can actually be this common underlying factor among the leading causes of death. In terms of how we study it, it gets incredibly complex, unfortunately, because there are many different markers of inflammation, but really we look at monocytes, cytokines, kind of various adhesion molecules, and then we have kind of non-specific markers such as CRP, and we have other specific markers that we look at, including interleukin-6 and various others. So there's a different, it's very dependent on what you're looking at, but typically you want to look at more than one. In terms of how we eat, if we think about foods that perhaps cause unfavorable postprandial responses, so postprandial meaning after we eat, you might then experience if you have repeated excessive kind of uncontrolled blood sugar and blood fat responses, this can kind of trigger kind of a chain of unfavorable metabolic effects that can perhaps lead to a variety of kind of negative short and long-term health outcomes over time, which could theoretically then lead into cardiometabolic diseases. So we're really looking at this, immune system response of something being quite unnoticeable, but then having this this long-term negative effect on your health. And actually the microbiome does play a role in this, potentially also leading to an unfavorable stable microbiome state. So in terms of the way that we talk about I mentioned earlier the gut barrier. So we see that Western diets, for example, containing typically higher fat and a lack of fiber, can change that kind of gut microbial composition. We see a reduction in those short-chain fatty acids producing bacteria, and we know that those short-chain fatty acids are really important to keep that integrity of the tight junctions. Whereas with the Western diets, we see kind of a loosening of the junctions. We see more pro-inflammatory cytokines moving into the body and inducing intestinal inflammation with this reduced mucus layer. However, on the other hand, with kind of prebiotic, which is typically fiber, which exert an effect on the microbiome and your health, and probiotic diets, which tend to be higher in fiber, we see an increase in short-chain fatty acid producing bacteria. We see increase in kind of tight junction proteins, in antimicrobial peptides, and also that maintenance of the mucus layer is really important. And that really helps to protect the integrity of the gut barrier, protecting from intestinal inflammation. There are also other mechanisms that have been suggested of how the microbiome interacts with inflammation. However, mechanistic studies are quite difficult, specifically in this area, and we have 20 minutes, so I'm not going to dive any further into those for now. What I would like to bring you towards is talking about PREDICT, which is the largest ongoing program to measure postprandial responses to food in nutrition science. And this is very much a collaboration between nutrition academia and tech, which has enabled us to be able to collect this really kind of high quality depth and breadth of data on a kind of unprecedented scale. So what we looked at is really very much trying to cover those questions of, what we eat, how we eat, and who we are, looking at a variety of exposures, including the microbiome, and then looking at the responses, which, are particularly topical to the conversation here, looking at inflammation as well. what we found was that we were able to identify microbes associated with favourable or unfavourable measures of health and diet. So we found this collection of we call them good microbes or bad microbes, and good being that we were able to see an association between good blood fat and blood sugar control. If you remember when I mentioned the previous slide that you want to have that an immediate effect that's not necessarily mediated by the microbiome, but can have a direct effect as well. If you have poor blood sugar control, that is then a contributing factor to that unhealthy metabolic cascade. We have lower levels of inflammation with these more beneficial microbes. We see lower levels of body fat and also lower risk of cardiovascular disease and type 2 diabetes. What we found as well was that not only were these good microbes associated with healthy measures of health, but they were also associated with diet. So specifically, healthy dietary patterns that were predominantly rich in healthy fiber plant-based foods, which we know are important for the microbiome. Likewise, on the other scale, we found these bad microbes or microbes that we are associated with the opposite side of higher inflammation and more negative health effects. So if we talk about diet, how can we modulate diet to support the gut microbiota and its involvement in immune health? And really the key core factor is going to be fiber. Fiber is what the fiber degrading microbes feed on to then produce the short chain fatty acids, which we know exert very strong health effects around the body. So we have specifically fermentable fibers, which are also classed as prebiotics. So it's really a substrate that is selectively utilised to confer a health benefit, and these typically tend to be inulin type fibers or galacto-oligosaccharides, and they're really known for their rapid fermentable capacity, and then subsequently they release these short chain fatty acids. So we see on the left here a variation of these different types of fibers. Particularly 95% of fiber typically is found in the cell wall and the cell structure. Likewise, if we think about what's interesting is also what your gut microbiome composition might be. So if you look to the right, they found, this is a study where they found that Prevotella copri was, which has previously been shown to be either positive or negatively influence on human health. And what they found was that some people who, if you didn't have Prevotella copri, then this likely modified the effect of fiber intake in alleviating chronic inflammation, here measured as CRP. So when we talk about fiber, it'd be hard not to talk about polyphenols as well. So these are really dietary compounds that are naturally found in a range of food and drinks. And typically they're found in plant foods, where actually it's the colour of each individual. So we get told to eat a rainbow, and really all the different colours that we find in these different foods are really from the polyphenols themselves. They're typically poorly absorbed, higher in the appetitoral tract, so they actually manage to get to the microbiome where they're selectively modulated by the gut microbiome. And there's extensive breakdown into these, in a series of metabolites that are then absorbable and exert all these different effects on health, including possibly reducing inflammation. So there's a recent study that came out in Cell that was run by collaborators of ours at Zoe, and what they looked at, it was a 17-week randomized prospective study, and they had, I think, 18 people in each arm, And they looked at a high fiber diet, but also the high fermented food diet, which is about 6 portions of fermented foods a day, which is a lot. It's not necessarily something that we could all strive to achieve, but I think it does show what's interesting is that the high fermented food diet was associated with increased microbial diversity and also decreased inflammatory signals and activity. And likely, you know, that's the combination between perhaps the, you know, prebiotic fibers that are present in quite a lot of these foods, if you think about kimchi, sauerkraut, but also possibly the live probiotic bacteria that are also present, because that's typical of the fermentation. These food products and drinks products are transformed by living bacteria and yeasts, and that's how they are fermented. There's also accumulating evidence indicating kind of a correlation between omega-3s and the gut microbiome. And we've seen in a number of studies that actually we can see this relationship, and perhaps there's also this is kind of a mediating effect from the gut microbiome related to you know, helping reduce inflammation. Not so much research and evidence as there is on fiber, but I think, you know, we know generally that omega-3 fatty acids tend to also have a direct effect, and then perhaps also we need to understand a little bit more about this area as well. So how can we further unravel these complex relationships? It's hard to communicate this enough in 20 minutes, but the diet and gut microbiota are, you know, inherently highly complex, and I perhaps should also add in there that also we talk about host health and immune system, very interrelated and difficult to assess. We really need kind of this precision, depth and breadth of data, and likewise we also need to understand the causality, you know, what is having a cause and effect rather than necessarily just an association. And really for that we need mechanistic studies. So really those optimal methods need to be incorporated, and also covariates considered, because, you know, the gut microbiome area of research has really kind of escalated at an astronomical rate in the last 20 years. And we're still kind of finding our feet of what is the gold standard approach. And we need to have, you know, very much a kind of consensus of how we can all move forwards together, so that we can have, you know, more homogeneity of outcomes. I do believe that nutrition has perhaps been seen as the poor man's science for too long, and I think, you know, we have an overwhelming amount of new techniques to explore the microbiome, but yes, you know, nutrition and diet research has been around for a long time. And there's many ways and tools that we can take from other areas of research and apply to diet to really model also the complexity of diet, while considering the fact that you know, we need to really be thinking about the specificity of the data and the precision of the data that we're collecting. Likewise, the microbiome is one piece of the puzzle. I do always like to remind people that, you know, food is, you know, tends to be, you know, absorbed kind of 90 to 95% of it before it reaches the colon, and there are direct effects from that as well. And lastly, microbial function is likely more important than microbial composition, and really thinking about those short chain fatty acids. I'd like to kind of thank everybody at Zoe particularly, and also the Predict study collaborators, and would love to answer any questions. Thank you.
Great, thank you so much, Dr. Leeming. And what I'm going to do is invite Dr. Sarah Berry to join us. So Dr. Berry, you may have seen her before on IFINS programs, but she is currently a reader at King's College London. She has over 20 years of experience leading more than 30 human nutrition studies on cardiometabolic health, and she's also the lead on Predict. So welcome, Dr. Berry. Dr. Berry is also on holiday, so an extra welcome for joining us. So we have a couple minutes for questions, and We had one that was about the markers of inflammation, and Dr. Leeming, you mentioned there are several. You might want to do more than one if you're trying to get a sense of the inflammatory process. So the question was really, what are the immune health related markers that show clinical relevance, if there is a subset or if we know about that?
So I think we can both answer that one, can't we, Sarah? So I think, well, CRP tends to be used in a hospital setting, but it's not necessarily something that we would always want to use in research. And I think, Sarah, I'll pass over to you because we use them in the PREDICT studies, and I know that you specifically selected the ones for a particular reason.
Yeah, so there's ones that have clinical relevance, and then there's ones that we know mechanistically are interesting to help us start to disentangle the mechanisms, the speed of action, etc. So CRP, as Emily said, is the one that has a lot of face validity in terms of being clinically meaningful, but we know that it's also highly variable. So if you have just a small cold, for example, it can go sky high. So whilst it's very good, I think, at a population level and to be able to detect really large excursions in inflammation, for the kind of research that we're doing, and I think for dietary inflammation immune research, we often need more sensitive markers that we know change within a time frame that we expect to see the diet change, but aren't sensitive necessarily to the kind of infection that we might not want to be measuring. So we typically look at markers such as IL-6 because we know that that's endothelial derived and it's highly sensitive to dietary modifications and can actually be modified in a really short period of time, such as, as quickly as 30 minutes, and certainly is useful when we're looking at postprandial responses. There's another measure that we're using a lot in our research called glyca. Glyca, we know, does change relatively quickly, is relatively stable within an individual, but is modified acutely by diet as well as chronically by diet. And glyca, we know of all the inflammatory markers now, which is an NMR, is picked up on an NMR panel, is one of the most strongly associated circulating measures with type 2 diabetes, cardiovascular disease, and all-cause mortality.
Great, thank you. So we had a question here that was about the point that function seems to be more important than composition. So the question is, what's the best way to quantify function as opposed to composition? And how can we tie these into health endpoints that are important?
Emily, that one's definitely for you, I think. You're the expert on that one.
So we're still, the jury's still out on what a healthy composition is, but I think tends to be that we're looking at, there's two things really. So I guess this comes down to function and also composition, where we're looking at specifically, butyrate producing bacteria, but very much producing enough butyrate because we know it does exert enough all these amazing health effects, which are important for your immune system, but also for other areas across your body. So it's very much, are we producing enough butyrate and are we producing enough short-chain fatty acids? And if we can see that versus specifically what composition of your microbes, then that is something that is positive and beneficial. One thing we can say from your microbes is that typically we're looking for anaerobic bacteria, so being in an environment where there is less oxygen, because that happens to be where your gut bugs love to thrive. And if we're introducing kind of an oxygen into the system, when we're getting this kind of proliferation of bugs and bacteria that like to use oxygen to grow and to flourish, those tend to be more pro-inflammatory bacteria, and typically those are in the kind of proteobacteria phylum. So what we do know on the flip side is that if you have a kind of unhealthy gut microbiota, this tends to be, used as a bit of a controversial phrase at dysbiosis, because again, how can we tell what an imbalance is if we don't know what healthy looks like? But we do see that there are certain kind of microbes in different disease states which are higher, that are not so good for health.
Gotcha, thank you. Okay, so because this was a really rapid mini webinar, we're basically out of time at this point, but we wanted to thank everyone for joining, and please look for future announcements about this series. Huge thank you to Doctors Leeming and Berry. And so we're going to close the public segment and have the speakers join the IFINS committee to dig further into the topic and understand where IFINS could possibly advance the science in this area. So thanks everyone, and have a great afternoon.
Thank you.